please confirm me in the chat box that whether you can see the slides or not, and then I will start. If you can see the slides, please do write in the chat box, and then I will start with the session. Okay, thank you for confirming. So last session, in the last class, from week 5 lecture session, we discussed only 2 questions. So let me start with that questions that were left in last class, and then we'll move to week 6 questions. So basically they are kind of same. Week 5 and week 6 both deals with enzyme kinetics and specific mechanisms of some enzymes. So both are actually linked, so it will be easier for us to discuss all these questions in today's session. So last time we did solve this question number 1 and question number 2 as well. So today we will begin from question number 3. Okay, fine. Those questions which I have already discussed in last session or similar question, I will go through them quickly, and new question I will give more time to that. Okay, so this is your first question of today's class. It states that which of the following is true about Michaelis-Menten kinetics. So in last session we have discussed in details about Michaelis-Menten kinetics and how to get all the equations for 3 different type of inhibition, etc. Fine. So the first question, it states that which of the following statements are true. Let us see. So the first statement is, it assumes that covalent binding occurs between the enzyme and the substrate. Is it true? If we do remember, the basic equation for Michaelis-Menten kinetics was, there is an enzyme plus substrate and they are forming a enzyme substrate complex, which were further resulting in the product formation. So we can clearly see that here is a reversible bond. That means the enzyme binds to the substrate in a reversible manner. So there is no covalent binding that is occurring. It is just a non-covalent type of interaction. That's why there is a reversible sign. So the first statement that has been stated here, that in Michaelis-Menten kinetics covalent binding occurs, it is a false statement. So you can clearly omit this first option. The second statement says, it always describes one molecule of the substrate binding with one molecule of enzyme. Again, so obviously if you look here in the kinetics equation, we can always see during the whole derivation we considered that one enzyme molecule is actually interacting with one substrate molecule. So the second statement that is given is actually a true statement. Coming to the third statement, option C, it states Km, or the Michaelis constant, is defined as the dissociation constant of the enzyme substrate complex. Okay, so we know it is our Ka and it is our Kd. Km has actually nothing to do with it. We can also write Ka as K1 and this as K minus 1 and this one as K2. Let me write it down, Ka or K1, Kd or K minus 1 and this one is our K2. If we do remember about Michaelis-Menten equation, so our Km value basically was Km equals to K minus 1 plus K2 divided by K1. But here in the third statement it says that Km is equal to the dissociation constant, that is basically your K minus 1. That is not true at all. So again our third option will be omitted. And the fourth option states Km, the Michaelis constant, is defined as the concentration of substrate at which enzyme is working at maximum velocity. And now if we do recall the graph that we draw for Michaelis-Menten kinetics, it is here V versus substrate concentration, and the graphs look something like this. So this one is your V0 maximum. And according to the definition of Km, it was, what was the definition for Km? One second, something like this. So this is your V max by 2. So you will go to the curve, and that particular substrate concentration will be Km, where your half of the maximum velocity is achieved. But in the fourth option, it is stating that the V0 max corresponding to that substrate concentration, the substrate concentration that corresponds to V0 max is this, is the Km. That is what is stated in your option number D. But we all know from the basic definition that this is wrong. Km is the substrate concentration at which half of the maximal velocity is reached. That is this particular concentration, not this one. So again, we can omit our fourth option as well. So the only option that was true among all the four statements that were given is option number B, that there is only one enzyme molecule is binding with the one substrate molecule. Moving to the next one. Okay. Question number 4 states, which of the following statements is true about competitive inhibitors? Let us see. The first statement, we all know about this already from the last class, that there are three types, basically competitive inhibition, non-competitive inhibition and uncompetitive inhibition. These were the three types of inhibition that we discussed in detail in last session as well. So first of all, the first statement is saying that competitive inhibition is a irreversible inhibition. Now again, I said this last time as well, that basically during the classification of inhibition, we can divide it in reversible and irreversible inhibition. Then out of reversible, there is competitive, uncompetitive and non-competitive inhibition. So obviously, competitive inhibitor is a type of reversible inhibition. So the statement that is given here, that it is irreversible mechanism, is actually false. So we can very easily omit the first option. Coming to the next one, the apparent Km decreases in the presence of inhibitor by a factor of alpha. Now, if you do remember the graph for competitive inhibition, so this is our normal graph in absence of inhibitor. Now, when we were adding inhibitor, initially the rate was slow, but after some point at high substrate concentration, they both reached the same V max. I am writing as Vm. So in both case, in presence of inhibitor as well, the V max was same for both the cases. And now what happened to the Km value then? So basically, this is your V max and half of V max will be somewhere around here. So without inhibitor, our Km value will be this, and as here Vmax is same, so Vmax by 2 will also be same. Now if we go to this graph with inhibitor, then our Km value comes something like this, writing as Km prime. So what we can see actually, that Km value increases in case of competitive inhibition when you add the inhibitor. So the second statement where it is said that Km decreases in presence of inhibitor is actually a wrong statement. So again, we are omitting the second option as well. Coming to the next one, it's saying that in presence of the competitive inhibitor, the Michaelis-Menten equation becomes this. The equation is written there. We all know that without inhibition, this alpha term is not there. When there is no inhibitor, the normal Michaelis-Menten equation is V0 equal to Vmax into S divided by S plus Km. But whenever you are adding competitive inhibitor, there is a term called alpha that is multiplied to Km, because now from the graph, just now we saw that Km prime, that is the modified Km value in presence of inhibitor, is always greater than normal Km, that is for without inhibitor. So that means alpha is always greater than 1, in case of competitive inhibition. So this statement that Michaelis-Menten equation changes by this parameter is actually a true statement. In presence of competitive inhibitor, the equation becomes, instead of Km only, the whole equation becomes alpha Km because of the increase of the Michaelis-Menten's constant value. So our third option that is given here is actually correct. Looking to the next one is the maximum velocity for the reaction decreases in the presence of a competitive inhibitor. Just now in the graph I have shown, because in case of competitive inhibitor, as we discussed in the last class as well, that in case of competitive inhibitors, the enzyme actually, or we should say the substrate and inhibitor both compete for the active site of the enzyme. So they are both competing for the same place. But whenever we reach or we increase the substrate concentration too much, that means even the inhibitor is present there, as substrate is now higher in number, so the probability of the substrate occupying that catalytic site will be more. So at a very high substrate concentration, we can actually omit the effect of inhibitors and achieve the maximum velocity. That is what is shown in here at the graph as well, that at high substrate concentration we can actually achieve Vmax. There is no decrease in Vmax in case of competitive inhibition. So the fourth statement, which states the maximum velocity of the reaction decreases in case of competitive inhibition, is actually false, because it stays actually the same. Just we saw from the graph. So the only option that was true out of these four was option number C. Okay. Moving to the next one, it states, okay, enzymes that differ in amino acid sequences but catalyze the same reactions, what are they called? There is a blank, you have to fill that. And the next thing is, and the molecule that acts directly on the enzyme to lower its catalytic activity is called what? So there are two things they are asking about. Number one is, one second. So the first one is an enzyme that has different amino acid sequence but are going for the same catalytic reaction. They can catalyze the same reaction. Suppose we are talking about hydrolysis. Both the enzyme A and B can hydrolyze, suppose a particular bond, but the amino acid sequence of A and B are different. And in number 2 they are asking, there is a molecule, suppose C, which directly interacts with the enzyme and then decreases the catalytic activity of the enzyme. So what will you call A, B and C? It was very easy. The first thing is, they are doing the same catalysis reaction. Although they are sequentially different, they are catalyzing same reaction. So what we can call them among these? Isoenzymes, because from definition we already know that coenzyme, apoenzyme, these are completely different things. Coenzymes or apoenzymes are basically, we should call, in case of holoenzyme, the protein part is called apoenzyme and the non-protein parts are sometimes called coenzyme depending on the type of bond. So they are completely different things. But whenever two enzymes catalyzes the same reaction, they are called isoenzymes. Why iso? Because although they are structurally or sequentially different, they are performing the same catalytic reaction. That's why in that case they are generally called isoenzymes. And the second option, where a molecule attaches directly to the enzyme and then decreases its catalytic activity. Obviously, what will be it? Whenever you can decrease the catalytic activity of an enzyme, it will be an inhibitor. Sorry, it was not writing. Okay. Obviously, it will be an inhibitor. So, our correct option for question number 5 was A and B, that the initial molecules that I was talking about, they were isoenzyme, and the third molecule, that is decreasing the catalytic activity, is actually an inhibitor molecule. Moving to the next one, it states, in the mechanism of lysozyme, aspartic acid 52, AsP52, stabilizes the oxonium ion through what type of interaction? Now, this is a question from your week 5 and week 6, not related to enzyme kinetics directly, but the mechanism of enzyme. So, please, if anybody can answer this question, they are welcome to write it down in the chat box, because it is very easy. If you do know the answer of this particular question, then write down what you think in the chat box. So, in case of lysozyme, AsP52 stabilizes the oxonium ion through what type of interaction? It is the question. So, first for that, I am already showing you the options since I don't see anybody. So, it is stating charge-charge interaction, but let us remember the whole mechanism through which lysozyme acts, and then we can come to the options one by one. So, first of all, what is a lysozyme? What does it do? It generally acts on, or it generally breaks the bacterial, what should I call that? One second. The bacterial lining, or the bacterial cell wall, whatever is there, it can break the bacterial wall. Lysozyme work is that, to break down the wall of the bacteria, cell wall of the bacteria. So, how does it act on that? So, the mechanism of lysozyme is shown here. So, if you do remember from the lecture that generally in case of the bacterial cell walls, you have your NAG-NAM units alternatively, and there are mainly two amino acid residue that are present in the active side of the lysozyme. One is glutamic acid 35, that is GLU35, that is being shown here, this GLU35, and another one is AsP52. These are the two amino acid residue that are present in the active site of the lysozyme. Now, if you do look at the mechanism, so basically glutamic or GLU35, it breaks the glycosidic bond, 1,4-glycosidic bond, for which you can see whenever this bond gets broken, so now we have a positive charge here. This is called the oxonium ion. Whenever this bond is getting broken by the presence of GLU35, there is a formation of this positive charge on oxygen, which is called oxonium ion. So, GLU35 has reacted, it has taken down this NAG moiety out, and there is our positive charge. But obviously this charge species needs to be stabilized by some counter interaction, because now initially there was no charge, but now you have generated a positive charge. So, for the next step to happen, you also need to stabilize that positive charge, otherwise it would never form. It is very obvious. And then comes the role for AsP52. What its role? It directly does not perform any kind of acid-base catalysis or anything. Like you can see in case of glutamic acid, it is directly involved. Okay, it is directly involved in taking out the, or giving out the H molecule, H plus molecule, and itself have become C-double-O minus. But in case of AsP52, it is not directly involved in any kind of bond breaking or making. It is reacting, or it is stabilizing the oxonium ion by a charge-charge interaction with its own negatively charged side chain. What does ASP or aspartic acid does have in its side chain? It has a COOH group or carboxyl group. Now this can deprotonate and can stabilize this oxonium ion at a pH where the reaction is happening. So what kind of interaction is this? This is obviously charge-charge interaction. So our options were charge-charge interaction, hydrophobic interaction, van der Waals and pipeline interaction. And for sure, if you do look at the mechanism, the correct answer for this question was charge-charge interaction. The entire other thing, see again in the next step as well, all the acid base catalysis is happening through glue 35, but AsP52 is only sitting there to stabilize the positive charge that has been formed. Once the positive charge has been removed, again it has no role. Okay, so our correct option was option number A. Moving to the next one, it states, suppose you are examining enzyme inhibition with different inhibitors. The efficiency of two distinct inhibitors for the same enzyme can be compared based on which of the following parameter. So what you are doing? So you have your enzyme E. Then you have your inhibitors IA and IB. These are the two inhibitors. Both can inhibit the same enzyme, but one can suppose do this more efficiently and another is less efficient. How can you distinct or make a distinction between them out of their capability of inhibiting the enzyme? What should be there? There are four options. Size of the enzyme, if I see, rate of the product formation, lowering the Ki value and higher the Ki value. First of all, from size of the enzyme, we cannot expect much because it actually depends on the catalytic site or orientation of the catalytic site. If it fits in the cavity, it does not matter what is the size of the enzyme. Say our cavity looks something like this, and one size of the enzyme is this and another enzyme is suppose this. So it does not matter as long as the inhibitor comes and fits in the catalytic site, it can inhibit the overall reaction. So size of the enzyme, that option is irrelevant. Rate of product in formation is nowhere relevant because you are dealing with enzyme and inhibitor, and the enzyme inhibitor complex never leads to the formation of product. So this fourth option is again irrelevant. Now only two options are remaining, that is what should be the value for Ki. If some inhibitor is a better inhibitor, then what should be the Ki value, higher or lower? If you now do remember the equation for don't forget this. This is the expression for Ki. Enzyme concentration into inhibitor concentration divided by enzyme inhibitor concentration. Now this is the expression for Ki. So we can see at the above there is enzyme and inhibitor concentration individually. So what should be the corresponding equation? The corresponding equation should look something like this. Plus I. Only then if you have to write the equilibrium constant for this equation, you will write enzyme concentration into inhibitor concentration divided by enzyme inhibitor concentration. So basically Ki is the dissociation of the enzyme inhibitor complex. So Ki value, what does it deal with? So it basically deals with the dissociation of EI. So if a EI complex dissociates very easily, that means the enzyme is again free. If the dissociation is very easy, then the enzyme will be easily removed from the inhibitor, and if the enzyme is free, then obviously it will go for the substrate and give us the product. That means our inhibition will be not much effective, obviously. So if our Ki value is low, that means the dissociation of the EI complex is very slow, because just look at this equation. This is the expression for our Ki. Now if Ki is very low, then this particular dissociation of EI complex will not happen much. In that case, that means lower the dissociation constant or Ki, that means not much breaking of EI complex. That means there will be very less free enzyme that can react with the substrate molecules. So if they cannot react with the substrate molecules, our overall reactivity will be decreased. So a lower Ki value generally means the EI complex is much more stable, it is not breaking down. That means the inhibitor is doing its job correctly. So the correct option should be in our case that lower the Ki value, better is the inhibition, because lower Ki means lower dissociation of enzyme inhibitor complex or EI complex. That means less enzyme or less free enzyme in the solution. So less free enzyme means less reaction with substrate and less product, and that is the job of inhibitor, and that means the inhibitor is working better. Okay, hoping this is clear. Moving to the next one. It states which of the following statements is true. So let us look at them. The first one is competitive inhibition is reversible and can be overcome by increasing the concentration of the substrate. Just in today's session we discussed this that competitive inhibition is indeed reversible, and at higher concentration, whenever we move to higher substrate concentration, we can actually achieve the Vmax as well. So by increasing substrate concentration, we can overcome the effect of the inhibitor. So our first option is obviously true. Coming to the next one, it states competitive inhibition is irreversible and cannot be overcome by increasing the concentration of the substrate. If this statement is true, this is just the opposite statement of option A. So obviously this is going to be wrong. First of all, it is not irreversible and we can overcome with the increasing substrate concentration. The third one is non-competitive inhibition is normally irreversible but can be overcome by increasing the concentration of the substrate. Now the first statement is very wrong. First of all, non-competitive inhibition is also a type of reversible inhibition, and again if you remember the graph for non-competitive inhibition from last session, it looks something like this, where even after increasing the substrate concentration, we cannot achieve the full maximum velocity. That means by increasing the substrate, you will not be able to overcome the effects of a non-competitive inhibitor. So our third option is again a wrong option. And the fourth one, it states competitive inhibition is reversible, fine, and cannot be overcome by increasing the concentration. Reversible part is fine, but this part is wrong. We can overcome inhibitor effect in case of competitive inhibitor by increasing the substrate. So again this part was wrong, so fourth statement was wrong as well. So the only correct option in case of question 8 was option number A. Fine. Moving to the next one. Which of the following statement is correct for the binding of a reversible non-competitive inhibitor to an enzyme? Okay, I will not spend much time in this because I have explained all this in the last session as well. So if we do remember the graph for non-competitive inhibition, so it is without inhibition, so V versus substrate concentration. Now when in case of non-competitive inhibitor, our graphs look something like this. So this is plus I and this is without inhibitor. So in that case, this was our Vmax in absence of inhibitor. Now in presence of inhibitor, our Vmax is this, which is much lower than the original one. So what happens to Vmax? In presence of non-competitive inhibitor, Vmax decreases. Now for the Km value, what happened? We have to find the Vmax by 2 value. So for without inhibition, our Km value was something like this. And again for non-competitive inhibition as well, our Km value is also coming here. Just by drawing you can understand that Km value does not change in case of non-competitive inhibitor. So what should be our correct option? Option number C, that there is a decrease in case of Vmax but Km remains constant. Moving to next one, it is saying enzyme catalytic activity is inhibited by a compound X. Upon increasing the concentration of the substrate, effect of X decreases. So what kind of compound will be X? First of all, it is an inhibitor because it is inhibiting the catalytic activity of the enzyme. Compound X obviously will be an inhibitor because it is decreasing the catalytic activity. Now again in the second line they are saying whenever you are increasing the substrate concentration very much, you can actually subside the effect of this inhibitor. So what kind of inhibition is this? Obviously competitive one. We just saw from a previous to previous question that in case of competitive ones, when you increase the substrate concentration, you can actually get rid of the inhibitory effect for competitive inhibitors. So the correct option should be this one. Okay, fine. Okay, this is a very interesting question. I will give you all some time to decide this. And please do write the answer of this particular question in the chat box. It states, from the options given, which one can best describe the type of inhibition or inhibitor shown in the figure? There are 4 options. This is your diagram shown on the right. Observe the diagram carefully and write down your answer in the chat box that what kind of inhibitor is the green one. So the green one is your inhibitor and the blue one is your substrate, that is already shown in the diagram. So what kind of inhibitor is the green one? There are 4 options: non-competitive, uncompetitive, feedback and allosteric. There are 4 options. If you have understood the types of different inhibitors, you would be able to answer this question. Please write it down. What do you think? That what should be the correct option for question number 11. I will give you all of you some time to think it out, but please answer this particular question. Any one answer from any of you? Just by looking at the diagram, you can actually answer this question. Fine. At least one of you, if anybody can answer this one. I am still waiting for all of your answers. I am just writing it down to further explain, but meanwhile, please try to answer this. Okay, I have one answer. Very good. Others also please try. Okay, fine. So let us look at the diagram and think about the inhibitors and look at the option and discuss. So the first one. First of all, you need to remember different types of inhibition. So to understand that, you have to remember their differences. Only then you can understand. So first mechanism that I have written is for competitive inhibition. Although this is not in the option, just for all of you to remember distinctly that what are the difference in different these kind of inhibitors, I am writing it. So in case of the competitive ones, the major difference is the inhibitor only reacts with the enzyme. It does not form any kind of ternary complex like ESI. It only reacts with enzyme. Fine. But in case of non-competitive inhibition, you can see the inhibitor can either react with enzyme or it can also react with the enzyme substrate complex, but ultimately the ternary complex will be formed. So in case of non-competitive, EI or ESI both can be formed. Okay, but in case of uncompetitive inhibition, the only way the enzyme can, the inhibitor can attach after the attachment of substrate. That after the formation of ES complex, the I can attach, thereby always resulting in the formation of a ternary complex or ESI complex. Also, now let us go back to this diagram. We can see that blue one is your substrate. Blue one is interacting and giving you the product. But this green one, if it was uncompetitive inhibition, then after this, basically this means this one is ES complex, this is your substrate, this is your enzyme, this is your inhibitor. So this one is your enzyme substrate complex. If it had been an uncompetitive inhibitor, then the addition of I would have come after the formation of this ES complex, thereby resulting in a ESI complex. That means the green one will attach after the attachment of the blue one. Is there something in this diagram where both blue and green are attached to this orange enzyme? No, we can obviously see there is no ternary complex. Both these intermediates, this one is ES and this one is EI. There is no presence of an ESI complex. That means it is not an uncompetitive inhibition. Fine. Now let's look at the non-competitive inhibition as well. In case of non-competitive inhibition also, we just saw the inhibitor can both bind enzyme and enzyme substrate complex, thereby also resulting in the formation of a ternary complex, that is ESI. But just we saw there is no such thing in the diagram where both the blue colored substrate and green colored inhibitor are attached to the orange enzyme. There is no such diagram or structure that is given there. So again, it cannot be a non-competitive inhibition as well. So we can rule out the first two options. I will come to the third option last. First, coming to the fourth one, allosteric inhibition. So what did we learn? We learn about allosteric inhibition. Allosteric inhibitor is some kind of inhibitor. They will, one second, let me draw it here. Suppose your enzyme looks like this. They will not bind to the catalytic site. What they will do, they will, bind somewhere else other than the catalytic site. This will result in some conformational change in the enzyme, for which the substrate molecules will no longer fit in the catalytic cavity. So they basically attach to some other side than the catalytic site and make some conformational change in the enzyme. Now let us look at the inhibitor here. When the green was, this is your inhibitor. So this green inhibitor, when it was attaching here, it is attaching to the catalytic site. It is not attaching any other site rather than the catalytic site, first of all. And after it is being released, because it is a reversible process, you can see after it is released, there is no such conformational change in the enzyme as well. This orange part is same after even attaching the enzyme. That means nothing like allosteric inhibition is going on. If it was happening, then the green one would have attached somewhere else, say somewhere else, or either here or somewhere here it could have attached, and this catalytic site or the conformation of the catalytic site would have been changed, something like this or this, something would have happened. But this is again not happening. So the fourth option, that is our allosteric inhibition, is also wrong. So only option left is feedback inhibition. And you have to remember this, the speciality of feedback inhibitor, it kind of looks like competitive inhibition, isn't it? Right? Because you can see only either ES complex is forming or EI complex is forming. So this one is our EI complex, this one is our ES complex. Enzyme is either binding with substrate or with the inhibitor. But the only difference with competitive inhibition is that this last step, in case of competitive inhibition, what happens? It gives you back the enzyme and your product. This is for competitive inhibition. But here what is happening for feedback inhibition, let me write it as F. The enzyme, after the enzyme substrate complex formation here, the product formation is happening. The enzyme is coming back to its original state, but the product that you are getting is itself an inhibitor for the enzyme. In case of competitive inhibition, it is a product, normal product written as P. But in case of feedback inhibition, the product itself is the inhibitor for your enzyme. So this sometimes happen to regulate the enzyme activity. Sometimes you do not want too much activity of a particular enzyme. That's why it happens that the product itself acts as an inhibitor. That means if too much product is forming, the product will itself block the enzyme and stop its activity. So always remember, here you can see that after reaction we are getting a green colored this one, which is actually an inhibitor as well. So that's why that is the basic difference between competitive and feedback inhibition. So our correct option for this particular one was option number C. And be very clear to remember all these different types of inhibition process. Okay. Okay, fine. Moving to next one. Okay. It is a math. It is very easy. I expect that all of you will be able to do that. It is saying in competitive inhibition, what happens to Vmax and Km if I is equals to Ki. So you have to remember the equation for competitive inhibition or the Michaelis-Menten equation and do this particular mathematics. I will give you some time. It will just take 1 minute to solve this question. Please do try it by yourself and then I will show you the solution for this. I can write down the equation here as well. That is the equation for your competitive inhibition. Please try to find out what would happen to Vmax. Try to solve it and then I will show in a minute or two. Okay, so fine. Let me show you the solution. So the correct, one second. Let me discard this. One second. So this is your solution. It is the equation what I have written for you. V0 is equal to Vmax into S, and in case of competitive inhibition, as Km gets increased, so there will be a factor alpha by which it will be multiplied. One second. By which it will be multiplied plus substrate concentration. And what is the equation for alpha, or what is the expression for alpha? I have already showed you that what is Ki. It is basically the dissociation constant for an EI complex, and I is your inhibitor concentration. This is the equation for alpha. So in this particular question, it is stated that I concentration or inhibitor concentration is equals to Ki. So obviously you will put here I is equal to Ki. So this will be 1 plus 1. So your alpha value would be 2. So if your alpha is 2, what will happen to Km? We all know in case of competitive inhibition, the modified Km or Km prime is basically alpha Km. That is without the inhibitor or unmodified Michaelis-Menten constant. So alpha value is 2. So the modified Michaelis-Menten constant will be 2 km. So this option, again we also know in case of competitive inhibition, Vmax does not change. So the correct option for this mathematical or numerical is supposed to be Vmax remains unchanged and Km increases to 2 km when I concentration is equals to Ki. Okay. Hope this is clear. Moving to the next one. It is stating a prosthetic group of a protein is a non-protein structure, that is what? So first of all, we have to know what is a prosthetic group. I have discussed this earlier as well. So we call these things as holoenzyme. That is, generally what is an enzyme? It is just a protein part, but whenever there is a protein and non-protein part involved, then they are called holoenzyme. That apart from protein, you also have your non-protein part. Now, these non-protein parts are generally known as cofactors. These cofactors can be of two types depending on the interaction that they have. Now, this protein and non-protein part, they either can have covalent interaction or they can have non-covalent interaction. These are the basic ways by which any two molecules can react covalently or non-covalently. Now, if the interaction between the protein and the non-protein part are non-covalent, then they are generally called as coenzymes. And if the interaction between the protein and non-protein parts are covalent, in that case they are called prosthetic groups. Prosthetic groups. So what should be the correct option then in our case? First option is ligand of the protein. It is not a ligand for the protein. First of all, it is reversibly associated with the protein. No, it is a permanent part of the protein actually. It is a part of the secondary structure of the protein. No, because it cannot have any secondary structure because they are non-protein parts, some kind of small organic molecules or metals. They cannot have secondary structures. And the only correct option is they are permanently associated with the protein because they are covalently added to the protein part. So that means they are permanently associated with the protein. So our correct option for prosthetic groups should be option number D. Moving to the next one. The reaction catalyzed by lysozyme is the hydrolysis of what? So obviously we just discussed this as well, that lysozyme actually hydrolyzes the bond between NAG-NAM, that is a 1,4-glycosidic bond. So if you do remember, just in some other previous question we discussed that it breaks down the bond between NAG and NAM, that is the 1,4-glycosidic bond in case of lysozyme. Okay. So I am moving to the next one. Fine. This is saying in the hydrolysis step for ribonuclease A. So now we are moving to ribonuclease A. So what is their role? They basically cleaves the RNA molecules. So in case of ribonuclease A, histidine 12 and histidine 119 respectively acts as what? These are the four options. So we already know that in case of ribonuclease A, there are two histidine moieties at the active site, his 12 and his 119. What do they act as in case of the hydrolysis step? Okay. Just give me a second. So let us go back here. So we know there are mainly two steps. The first step is, I don't know if I have a, okay. So let me just show it with this mechanism part, then it will be easy. So therefore, there is basically two steps in the mechanism of ribonuclease A. The first one is cyclization and the second one is hydrolysis. They are here asking us about the hydrolysis step, which comes after the cyclization step. So basically the role of histidine 12 and histidine 119 are reversed in these two steps. That histidine which acts as a base in case of cyclization acts as an acid in case of hydrolysis. Just their roles get reversed. Now for us, the question is about the hydrolysis step. So this is your hydrolysis step. after the cyclization has been happened, this is the hydrolysis step. So what is happening here? This is your histidine 119 residue. That is taking up a proton from water molecule. Why? So that we can generate a hydroxyl ion which can then attack this P double bond O center. Okay. So what it is histidine 119 is doing? It is taking up a proton from water molecule. So whenever something takes a proton molecule from other source, what do we call them? It is taking up proton molecules. That means it is acting as a base because it is taking up H plus. And what is histidine 12 doing here? After this hydroxyl ion attacks here, so this bond is breaking and becoming O minus. Now it will find something to be stabilized. What it can see? There is a hydrogen atom for histidine 12 which it can abstract and form OH, right? So what histidine 12 is doing here? It is giving its H plus to this O minus. So it is giving away H plus molecules. So whenever some molecule gives away H plus ions, that means it is acting as acid because it is giving H plus. So that means in the hydrolysis step, this particular histidine 12 residue acts as an acid and this histidine 119 residue acts as a base. So for hydrolysis step, our correct option was option A. Histidine 119 as a base and histidine 12 as an acid. And if you look at the cyclization step, the role will be reversed. Histidine 119 will act as an acid and histidine 12 will act as a base. Okay. Moving to the next one. So it states, in a plot of 1 by V against 1 by S for an enzyme catalyzed reaction, the presence of a non-competitive inhibitor will alter what? So I will go through this very quickly because I have discussed this numerous times. So this is your 1 by V versus 1 by substrate concentration. This is without inhibitor, and in case of non-competitive inhibitor, it will look something like this in presence of plus I. So what does it do? It alters the intercept of the y-axis or 1 by v-axis. It was initial before inhibitor and it is after inhibitor. So the intercept has been changed. That means our correct option should be C, that it changes the intercept on 1 by v-axis. Moving to next one, it states the above reaction is catalyzed by succinate dehydrogenase. This particular reaction is catalyzed by succinate dehydrogenase. Okay. Now you get this particular product. Then they are saying this is another X molecule. Now you are giving succinate dehydrogenase or adding succinate dehydrogenase to this X molecule. Then what will be the product? After reaction of X molecule with succinate dehydrogenase, what will be the product? These are your options. Try to answer it, and then if you anybody from you can answer this question, write it in the chat box and then I will tell you. From the name itself, it is very much clear, dehydrogenase, and you can see it in action that it is basically removing hydrogen atoms from these molecules and forming a double bond. Okay, so fine. We will come to the options later. First, let us see what is SD doing. Fine. Now, if we carefully look what is SD actually doing here, it is abstracting the alpha hydrogens. So, this is your alpha hydrogens. This is your carbonyl groups and this is your alpha hydrogen. There are two alpha hydrogen. Here are also two alpha hydrogen molecules. So what succinate dehydrogenase is doing is abstracting two hydrogen atoms or two alpha hydrogen atoms, thereby forming a double bond in between them. Now if the same thing has to happen in case of X molecule, so this is the structure. So what are the alpha hydrogens in this case? This is one carbonyl. So this is our two alpha hydrogen, and this is again another carbonyl. This is another two alpha hydrogen atoms. But these ones are not alpha hydrogen, these are beta hydrogens. Now SD is only abstracting alpha hydrogens. Now if it had to act on this X molecule, so it will take up one hydrogen from here, one alpha hydrogen, and another one alpha hydrogen from here. If that happens, now in this case, because these two alpha carbons were next to each other, after abstraction of the hydrogen, you can form a double bond in between them. But in this case or this structure, there is a CH2 group which is hampering this reaction to happen, because even if you take up the alpha hydrogen atoms, the C alpha, these are your C alpha, they are not next to each other. There is a C beta in between. So the double bond that should have been formed between the C alpha is no longer can happen. That will not be stable even if it happens. So that's why even if you add succinate dehydrogenase in this particular molecule, it will not result in any kind of product. Why? Because it only will abstract alpha hydrogen, and even if it does, it is not leading to the formation of any kind of stable double bonds. So it will not act on this particular X molecule. Yes, who answered that? Yes, Akshara, your option was absolutely correct. Okay, moving to the next one. It is saying in the given diagram, the enzyme inhibitor interaction is shown among two paths, A and B. Suppose I am saying this is your A and this is your B. Indicate the probable type of interaction in each path. The respective shapes are indicative of the activity of each species. That means same representation indicates same activity. Now let us look here. It is our enzyme, it is our inhibitor complex. In both path A and B, the enzyme inhibitor complex looks same. Now after this A process, the inhibitor looks same here in case of A. The enzyme also looks same, just like the first ones, that at the beginning of the reaction. Let us look at pathway B. Enzyme inhibitor complex looks same, it is fine. But after pathway B, you can see the inhibitor looks same, just like the beginning of the reaction. But the shape of the enzyme now has been changed. That means the activity of the enzyme has been changed, because that is what is written in the question, that the shapes are indicative of the activity. Now it has become round. That means, and also you can see from the notation as well, it is written as E prime. That means there is a change or conformational change that has been happened to the enzyme. So now they are asking what kind of possible interaction might have happened in case of pathway A and pathway B. So let us look at A. Obviously in case of A, no such change has happened. So we can obviously think that whatever interaction the enzyme had with the inhibitor was a non-covalent type. Only in that case it will not lead to any kind of structural or conformational change that can alter the activity of the enzyme. So that means in pathway A, non-covalent interactions were actually happening. But in case of pathway B, because there is a change in the shape and the catalytic activity, that means some configurational change has been happened there, which is only possible if the inhibitor has reacted in the or attached to the enzyme in a covalent manner. Some kind of reaction has been happened via formation of covalent bonds which has altered the conformation of the enzyme. So the correct option for that should be option D. Our pathway A was non-covalent and pathway B was covalent. Fine. Moving to the next one. It is stating salivary amylase is an enzyme in humans that breaks down starch. Fine. So I am just writing it as SA. The role of it is to break down starch molecules. The optimum pH for this reaction is 6.7, and it is also known that this enzyme is associated with calcium 2+. In conditions lacking calcium, the enzyme would be referred to as A. So we can already say that there are two parts. One is the salivary amylase and another is the calcium 2+. Whenever you remove the calcium 2+, you are only having SA, that is referred as A in the question. And the rate of this reaction would not be affected by the condition depicted as B. What is A and B? So first of all we need to understand that whenever some kind of metal ion is associated with the enzyme, that means their overall interaction or their overall combination will be known as holo enzyme, because now we have our protein part SA and non-protein part calcium 2+. So if you remove calcium 2+, then the enzyme is referred as A. So if you remove the non-protein part, what are they called? If you cut down the non-protein part from holo enzyme, they are known as apoenzyme. So obviously from the options already you can sort out that either option B or D should be correct, because in absence of calcium 2+, we should call the enzyme as apoenzyme. Together they will be holo enzyme. Again they are saying the rate of this reaction would not be affected by the condition depicted as B. So obviously you know the pH of the reaction that is necessary, that is 6.7, that is necessary for the catalytic site of SA to do the reaction. But in absence of calcium 2+ as well, the enzyme would function. The pH is actually responsible for some amino acid site groups that are present in SA. So even if you remove calcium 2+ and after that you maintain the pH of the reaction at 6.7, it would not affect the overall condition of this reaction. So as long as you maintain the pH 6.7, the catalytic activity of SA would go on. In presence of calcium 2+, obviously it will be better. But even in absence of that, if you maintain the pH of the reaction at 6.7, the overall conditions of the reaction or rate of the reaction would not be affected. So the correct option for this correct answer for this particular question should have been option number B, that A is an apoenzyme, and if you maintain the correct pH, it would not alter the reaction rate of the reaction or overall condition of the reaction. Okay, moving to next one. Again, it is showing a diagram. In gist, if I have to read it, it is saying that there is enzyme denoted as ES, denoted as S, substrate denoted as S, and I for inhibitor in this particular diagram. For the depicted enzyme substrate interaction, the inhibitor can either bind with the E or ES. Based on the depicted enzyme substrate interaction, which of the inhibitory process is going on. So I have discussed this distinctly many times, that whenever an inhibitor can form both EI and ESI complex, they must be non-competitive inhibitors. So our yes, absolutely, all of you are correct. So the correct option for these questions is option number C, non-competitive inhibition. Moving to next one. Fine. Again, this is sorry. VSI. Okay. Okay. Again, it is saying that identify the species unique to this kind of inhibition. So, what is the identifiable species? Because there is a formation of ternary complex. This separates the non-competitive inhibition from competitive ones. So, the ternary complex formation, absolutely option A is correct. The ternary complex formation is the unique species of this particular inhibition process. Okay. Again, let us look at this. So, you have to match column A with column B. There are 4 options in column A and many more in B. So, you have to choose them according to their mode of action. Now, if you do remember, in the last session, week 5's first question was, how does insecticides works? What are insecticides? And we said that insecticides are generally choline esterase inhibitors. So you already know this one because in last class we have solved this, that insecticides are generally choline esterase inhibitor. Now look at the other ones. Nerve gases. They are extremely dangerous because of what? Because nerve gases are able to bind irreversibly with an enzyme. So that means it can completely stop a catalytic activity that is happening. If it is irreversibly bound, there is no more chance that the substrate molecule come and bind to that enzyme anymore, and the catalytic reaction is completely stopped. That's why it is dangerous. Okay, I have already 2 options, can see. Next one, say antibiotics. Obviously, if an antibiotics has to work, it should inhibit the enzymes by affecting bacterial metabolisms. Sorry. If an antibiotics has to work, obviously it has to inhibit the enzymes that are responsible for bacterial metabolism. If it can stop the bacterial metabolism, then it can stop their spreading or their replication. So that means antibiotics do that job of by inhibiting the enzyme that are responsible for bacterial metabolisms. Okay. And the 4th option is heavy metal poisoning. Now what kind of these are like heavy metal are like mercury, cadmium, like this. So if you remember from your class 9 or 10 classes, that mercury, cadmium, these are called as soft metals. So they will be finding soft interaction very much favorable. So what they will do, so now if you remember, in case of amino acid, we generally have carbon, nitrogen, hydrogen, oxygen. These are known as hard centers. They are not regarded as soft ones. The only soft center that you can find in case of amino acid and thereby protein is the sulfur related something. Sulfur is referred as a soft center. So what they will be find where? Obviously cysteine residues, SH, and because in case of cysteine it is SH, it is very much reactive. So they are very much reactive to the soft metal centers like Hg2 or something, where they can have a soft interaction and have a good binding. So most of the time the heavy metal poisonings that do happen by irreversible enzyme inhibition through binding with the cysteine residues. So if you now we have all the options matched correctly. So now if we look at the option, our correct option should be option number C. Okay. How many questions are we left in this session? Just give me a second. Not many, I guess. Okay, we'll quickly go through this. Not many. Fine. So the next question is, you already know this. The diagram shows the working of the enzyme dash by the dash mechanism. What does A and B stands for? So obviously you know that GLU35 and ASP52 is nothing but your lysozyme. They are present in the active sites of the lysozyme, and this mechanism that is shown here, that is the formation of the oxonium ion and stabilization by AsP52, and then again acid base catalysis by GLU35, is known as Phillips mechanism. So the correct option, one second, for this is the enzyme that we are talking about here is lysozyme, and this particular mechanism is called Phillips mechanism. Moving to the next one, we also discussed this one. The enzyme whose mechanism is shown is specific for catalyzing what? We already discussed that this is a lysozyme enzyme which is responsible for breaking down the bacterial cell walls. So bacterial cell walls has NAM-NAC alternatively. So what lysozyme does is break the glycosidic bond that is shown here, break the glycosidic bond that is here between NAM and NAG, and the conformation of the bond is beta 1,4. So the correct option for lysozyme. So first of all, our correct enzyme that is working is lysozyme, which does break this beta 1,4 glycosidic bond. So the correct option for question number 24 should be A. Fine. Again, this is a very easy one. Given the Lineweaver plots of enzyme inhibition, state which graph represents which process. So obviously starting from this one, what kind of process is this? We can already see that the Km value is, this slope is minus 1 by Km. So the Km value has decreased but the slope is constant. That means Vmax has also decreased. So whenever Km and Vmax both are decreasing and the slope is remaining constant, that means it is a type of uncompetitive inhibition. Now in this case Vmax is changing, that means Vmax has been decreased but the Km value remain constant. Whenever this happens, this means this is a non-competitive inhibition. And the last one where Vmax remain constant but your Km value increase, this means it is a competitive inhibition. So our correct option should be A uncompetitive, B non-competitive and C competitive. So option number D. Yes, who answered it? Akshara, yes, you are correct. Okay, this is the next question. Based on the type of inhibition occurring, identify the correct changes in case of Km and Vmax. We just discussed in the last one as well. So obviously this one is uncompetitive where Km value decreases and Vmax also decrease. Now this one is non-competitive where Km value remains constant but Vmax decrease, and this one is competitive one where Km increase, Vmax remain constant. So let us look at the option in the next page. So yes, the correct option should have been A. For the first uncompetitive one, both decreases. For non-competitive one, Km constant but Vm decreases. For competitive one, Km increase but Vm remain constant. Okay. This is the same thing, I guess. Again, it is saying that K cat or turnover number, what will happen to it in case of the three different inhibition process. Now, we all know that this is the equation for K cat. What does it depend on? Vmax. The total enzyme concentration is not going to change for all the inhibition. So, obviously, for the first, that is uncompetitive one, since Vmax decreases, so there should be a decrease in K cat as well. In the second one, that is non-competitive one, where also the Vmax decreases, so K cat should also decrease. And in the third case, that is the competitive one, where Vmax remains same, so that means it should remain constant. So our correct option should be yes, absolutely, all of you answered right. The correct answer should be option number B. A decreases, B decreases, but C remain constant. Okay. Fine. This again states that a researcher synthesizes a certain compound known as Mn and claims that this particular compound can cure cancer. When he proceeds to publish this work in a journal, his claim is promptly rejected. From the given information, identify why his claim was rejected. Okay, so these are the four things that he reported for his compound Mn. So this was your compound and he reported that Mn has high biocompatibility, high solubility, non-toxicity and this Ki value. Now let us see what is the problem with his particular compound. First of all, high biocompatibility. Whenever you are saying something about a drug molecule, suppose it can cure cancer, mean it is some kind of drug molecule that you have to put inside your body. Biocompatibility is the thing that means that whenever you put that particular compound inside your body, it does not generate any kind of unwanted reactions. That means biocompatibility, to say in a easy way. Whenever you put it in your body or it goes inside your body, it does not react in a bad way. No unwanted reaction is happening inside your body. It is compatible with your system. So obviously high biocompatibility is a good thing, that is absolutely necessary for any drug molecules. So obviously this particular point is valid, so they cannot reject that compound based on high biocompatibility, because it is very much required. So the first option that is given, that high biocompatibility is not good for curing cancer, is absolutely wrong. If it has to cure cancer, it has to go to our body, so it has to be biocompatible. So the first option is total no. The second option is high solubility. Obviously the main problem with the drug molecules is the solubility, that if you are injecting something specially, say intravenously, that means the compound is directly going to your blood. And what does our blood have? It is generally based on water. Our whole body is basically based on water. So if your drug molecule is non-soluble in the blood or the water media, then how can it reach the desired sites or the cell molecules? The cells. So it is a very important factor that the compound has to be highly soluble, and he is reporting that Mn is highly soluble. So this should have been not a problem for the reviewing committee of the journal to completely discard his claim. So again, this was not the problem. High solubility is a desired thing, so this was not a problem with that compound. The next one is non-toxicity. Obviously, it has to be non-toxic. What does non-toxicity means? So, you have invented a drug somehow and it works on the cancerous cells. Very good. But whenever a person is having cancers, that means he or she has both healthy cells and cancerous cells. It is very fine that the drug is killing your cancerous cell, that is absolutely fine. But if it also starts to kill the healthy cells of your body, then it is doing more harm than good, because you need your healthy cells to stay alive. So non-toxic means it is non-toxic, or it is not killing your healthy body cells. It is only killing the cancerous cell. So again, non-toxicity is a necessary criteria for any kind of drug or compound to be injected in case of our body. So it does not affect the activities of our healthy cells. So again, non-toxicity thing that is been take care about the fourth point. It is highly necessary for it to be non-toxic. So again, this thing should have been not a problem for this compound to be rejected. This is a necessary point. They shouldn't have been rejected. The only point remains is the Ki value. So what is the problem with the Ki value? If we look at it, the Ki value is 5.7 into 10 to the power 8 molar. That is an extremely high value. And what did we discuss about Ki value? If we think in terms of enzyme, Ki was dissociation of the enzyme inhibitor complex. So he designed a thing, Mn, compound called Mn, which is actually an inhibitor and say somehow blocks the enzyme that are present in the cancerous cell and stops the proliferation of the cancerous cell. But to do that, if it is inhibiting the some kind of regulatory enzyme in case of cancerous cell, then this inhibition or inhibitory complex that has been formed has to be very stable, because only if this Mn compound can stop the activity of the enzyme, only then it can stop the cancerous cells to further proliferate. Okay. But if the enzyme inhibitor complex is very easily dissociated, that means it is not effective in demeaning the catalytic activity of that enzyme present in the cancerous cell. So yes, the Ki value is very high in this case, which is depicting that most of the time the inhibitor is actually not doing its job properly and not blocking the enzyme at the cancerous site. That means it will not be able to properly act, the cancerous cell will still proliferate. This is why by looking at the Ki value, that although the biocompatibility, the solubility, the non-toxicity, everything was fine, but it was unable to stop the enzymatic activity that was happening at the cancerous cells. That is why his claim was rejected from the journal. So our correct option is option C, exceedingly high Ki value. That is why they have rejected this Mn compound. Okay. Moving to next one. We all know this. It is saying the given structure is an integral part of two very important biological molecules. We can see this. This is basically porphyrin. So why it is important? Porphyrin is present in case of hemoglobin and myoglobin. What is myoglobin? It is a monomeric protein. And what is hemoglobin? It is a tetrameric protein. So our correct option should be option number C, that this given biological molecule, that is porphyrin, is actually very important for monomeric myoglobin and tetrameric hemoglobin molecule. Okay, let us move to the next one. It states the structure coordinates with the metal ions M2 plus for their functioning in the human blood. So here is your M. So for this whole structure to function, it has to react with this M2 plus. When X coordinates with M, the structure is referred to as Y. M and Y are what? So M is something, this kind of metal, we don't know what is M. One question is what is M, and another question is what is this framework that is outside M, that is holding the M molecule basically. So what is this framework? That is denoted as Y. So you all know that in case of human blood, what is it? It is hemoglobin that is found. So the Y, or that outside framework, is basically porphyrin, and in case of hemoglobin, what remains as the metal inside? It is nothing but Fe2 plus. So the correct option for this, and together, I mean after the Fe2 plus is attached with porphyrin, the entire structure, when X coordinates with M, what is this entire structure called? It is known as heme. So our metal is Fe2 plus, the external structure or the external coordinates, this is basically porphyrin, and whenever you add Fe2 plus with porphyrin, then you get your heme molecule. So the correct option for this is M is Fe, the X coordinates, these are porphyrins, and overall the Y is heme. Okay, I think this is the last one from this. Very good. So yeah, we do have another 15 minutes or so. Let us continue with the questions of week 6 then. Okay, fine. Since we do have some time, we can continue some questions of week 6. So these are the questions that are directly taken from your week 6 lecture material, where we learned about different type of proteases, defined enzyme degradation of peptide chain to get the sequence, etc. the questions are regarding these things. So question number 1 from week 6 states, serine proteases are the largest class of mammalian proteases. Why they are called so? Why they are called serine proteases? So you all know that already. So these are your 4 options actually. The first is they cleave peptide bonds following serine residues in a protein. They have a serine residue at their active sites. Serine inhibits their functioning. And the fourth one is they destroy the structural properties of serine. But you all know why we call them serine protease? Because, one second, just give me a second. Because if this is your protease enzyme, then there is a serine molecule that is present at the catalytic site, which is why they are classified as serine proteases. I mean, there are different class of proteases molecule, but among them which have serine at their catalytic site or active site, only they are termed as serine proteases. So our correct option is that their name comes from this because serine resides on the active site. Moving to the next one. It says proteolytic cleavage or peptide chain cleavage is the breakdown of proteins into smaller polypeptides or amino acids. Okay. So the peptide bonds are very stable. If it is not catalyzed, it may take up to 100 of years to occur naturally. We all know that because we all are made of proteins, and if it has to naturally, protein could have been very easily broken, or the amide bonds could have been very easily broken, then we would not be alive by now. So we all know these facts already. Then they are saying there are two major methods for catalyzing the peptide chain cleavage, enzymatic and non-enzymatic. So basically you have many peptide bonds in your protein. Naturally you cannot break it easily. It will take very long time. So you need to use catalyst molecule to fast up reaction, to fasten the reaction basically. So what will be this catalyst? There will be different kind of enzyme. Now in case of peptide chains, you can cleave these amide bonds either by using enzymes, that is a enzymatic pathway, or sometimes they can also be cleaved by some organic molecules as well, which is known as non-enzymatic pathways. So below are some of the examples. You have to choose which is the correct example for chemical or non-enzymatic cleavage and followed by enzymatic cleaving mechanism of peptide chains. So two options are given, subsequently A and B. A1 is non-enzymatic and the B1 is enzymatic. From this option you have to choose. Now first of all, if you look the correct options that can cleave enzymes among these, first of all we have to find this out. So the molecules that are mentioned here, only trypsin, that is an enzyme, and CNBR, they can break amide bonds. Okay, they can cleave peptide chains. This is their role. Tyrosine and arginine are amino acid. They cannot break peptide bonds, obviously. And CNB is not the compound that we are talking about. CNBR is the reagent that can break the peptide bonds. Now, out of them, you obviously know CNBR is not an enzyme. It is an organic molecule. So, the cleavage by CNBR will be regarded as non-enzymatic or chemical pathway. So our non-enzymatic pathway, we can write down, it is CNBR. And in case of enzymatic pathway, we have our trypsin. Trypsin is an enzyme. So if we are using trypsin, we can call it a enzymatic pathway. So our correct option was, for chemical it will be CNBR, and for non-chemical or enzymatic it will be trypsin. Fine. Moving to next one, it is saying Edman degradation. Already you know, it is a process developed by Edman that helps in sequencing the amino acids of a peptide. In this method, what happens? The amino terminal is clipped one by one. So if this is your peptide change, this is your N terminal, this is your C terminal. So one by one your N terminal amino acids starts to come out. So first N terminal will come out and you will end up with a smaller peptide change. Here again this is your N terminal, this is your C terminal. Again this N terminal part will come down in the next step and you will end up with another shorter peptide chain, and this will go on. This is for Edman degradation. So it is saying which of the following compound is not involved in case of Edman degradation. There are 4 options. Which of them are not involved in case of Edman degradation? For answering this question, excuse me, let me just remove it down. Yeah, if I discard it, then it will be easier for all of you to see it. So this is the basic mechanism for Edman degradation. So basically what we do in case of Edman degradation, we use phenyl isothiocyanate okay. So we use, don't use the pen. We use phenyl isothiocyanate, also known as PITC. So this is our phenyl isothiocyanate. At the first step happens at a slightly basic condition. So in our Edman degradation, phenyl isothiocyanate or PITC is involved. Then in the next step we require H plus or a slightly acidic medium. So obviously if we look here, CH3COOH is an acid, so it might be required or it will be required in very small amount in case of Edman degradation, because you require very small amount of H plus, a little H plus. So a very small amount of trifluoroacetic acid will also be required in case of Edman degradation. So this is also required. From the mechanism you can tell. And then we have our formation of phenyl thiocarbonyl or phenyl thiohydrantoin, that is this PTH moiety. So PITC, PTH and acid all are involved in case of Edman degradation. The only thing that is, or compound that is not involved in case of Edman degradation is 1,fluoro-2,4-dinitrobenzene or FDNB. Okay. Okay. Moving to the next question, we have 5 minutes, I guess. Okay, we'll continue. Which of the following are serine proteases? So serine proteases we already discussed. It is a class of proteases that have serine molecules at the active site of it. So among these, which are the types of serine proteases? So if you do remember the lecture material, chymotrypsin and trypsin both are a type of serine proteases, and we learned the mechanism of chymotrypsin in details. So the correct option for a question number 4 should be A, where it states chymotrypsin and trypsin are serine proteases. ATP synthase is not a type. It's, first of all, it is not a protease, and obviously it is not a serine protease. Moving to next question. Which of the following is true about myoglobin? One second. Is it even audible? Okay, I think my connection. Can all of you see it now? I think my net connection got interrupted, and probably all of you thought that I have left. One second. Okay, sorry for that. My net internet connection got interrupted, and all of you must have thought that the session was over. Very sorry for that. Let me present it again. Just 5 minutes more, and then I will actually end the session. Those who have left the session already, they can just look at the YouTube videos to see the solution. Unfortunately, I did not realize when the internet connection was over, and I kept showing the questions and everything. You can look that part in that YouTube video because it was being recorded, so you can see that part from the YouTube itself. Okay, so just one or two questions more, and then we will actually end this class. So the fifth question that we are talking about, it states that which of the following is true about myoglobin? First of all, we know that there are two proteins that are one second. There are two proteins that are involved in case of any interaction with oxygen. One is hemoglobin and one is myoglobin. Out of them, we know hemoglobin is a tetrameric protein that has 4 subunits, but in case of myoglobin, it has only 1 unit. So obviously, if hemoglobin does have 4 subunits, it can bind up to 4 oxygen molecule as well, and in case of myoglobin, as it only has 1 unit, it can bind only 1 oxygen molecule. So again, hemoglobin is found in our RBC or red blood cells, that means in our blood. This means again, blood, what does blood do? They bind with the oxygen and transport it to different parts of our body. So hemoglobin's job is to transport the oxygen molecules, but in case of myoglobin, they are found in case of muscles and tissues where they store the oxygen molecules for future use. Okay, they basically store the oxygen molecules that are being transported by the hemoglobins. So in this particular question, our correct option should be myoglobin is monomeric and it majorly function as a oxygen storage protein. And if it was case for hemoglobin, our correct answer should have been that hemoglobin is a tetrameric protein and it mainly function as oxygen transport protein. Okay, so fine. I I think just after this question we will stop. So this states, in the given reaction scheme, the starting material is p-nitrophenol acetate. At the end of the reaction, the product obtained P is yellow in color. If Z is an enzyme, then what is Z and what is the function in the given reaction scheme? So we all know already this particular equation. Very sorry. This particular equation is, one second, it's mainly used for chymotrypsin. Chymotrypsin generally breaks the carboxylic side chain of a hydrophobic side group amino acid residues. Amino acids that have hydrophobic groups in their side chains, chymotrypsin targets those peptide bonds and breaks them from the carboxyl side or the C-terminal side. Now, chymotrypsin can also act for the cleavage of ester bonds. What kind of bond is breaking here? A ester bond has been broken here. That is, a cleavage of this ester linkage is happening in this case. So, chymotrypsin can also function as ester cleaving molecules, and in this case, by this way we can also check the activity of chymotrypsin. So whatever happens when they do react with this para-nitrophenyl acetate, they are this para-nitrophenyl acetate, they are colorless molecules. So if you add chymotrypsin to this colorless compound, then we end up with the cleavage of this ester bond and formation of this particular compound, that is para-nitrophenol, which is in equilibrium with the higher, basically the H plus got removed and you got your O minus or alkoxy group here. So whenever this deprotonation takes place, this particular deprotonated para-nitrophenol has a distinct yellow color. Now by generation of this yellow color, you can actually check the activity of chymotrypsin. Say you have chymotrypsin for your in your lab for a very long time and you don't know whether this enzyme is still active or it has loses its activity. So for that you can simply take para-nitrophenyl acetate and then add your enzyme or chymotrypsin in it. If it changes color, that means it is still successfully cleaving the ester bonds. It still has its activity intact and you are getting the color for the alkoxy ion or the O minus ion that is forming from para-nitrophenol. And if the yellow color is not happening, that means the chymotrypsin has lost its enzymatic or catalytic activity and you can no longer use it. So the correct option for question number 6 is that in this particular reaction that is shown here, Z is actually chymotrypsin, which is functioning by cleaving the ester linkage that is present in case of para-nitrophenyl acetate. Okay. So with this we will end. I will upload the video. Let me just stop the recording. And the rest of the question we will again discuss in next class. So thank you very much all of you to listen to this session.